We'll move on to the next speaker so long, which is Leah. She'll be presenting work on Immunoinformatics approach to the design of a peptide-based vaccine against malaria.
Hi, can you hear me?
Yes, we can. Go ahead.
And you can see my screen?
Yes.
Okay, perfect. Good afternoon, everyone. I'm Leah Maharaj. I'm a PhD student at the University of KwaZulu-Natal in the discipline of genetics. And I'm presenting my project, which is titled The Immunoinformatics Approach for Peptide-Based Vaccine Design Against Malaria. It's a common misconception that malaria is a disease of the past. But who has reported that control measures have stalled over the years? It's still occurring in the Southern Hemisphere, affecting a large proportion of Africa. To approach elimination, vaccine design is prioritized. This study looked into vaccine design using immunoinformatics tools. The research problem being addressed is the fact that malaria vaccines have thus far had limited efficacy. An in silico approach will save time and money while giving an idea of how effective we can expect the vaccine to be. The aims are to design B and T cell epitopes and see if these stimulate the major histocompatibility complex. This will be done by designing CD4, CD8, and B cell epitopes with online servers, determine population coverage of the HLA allele, and to conduct molecular docking. This is an overview of the protocol that was used. So I pulled all NCBI sequences for Plasmodium falciparum, which is the causative agent of malaria, and selected regions that align to CD8 and CD4 reference sequences. Those that showed antigenic and transmembrane properties were chosen. CD8 and CD4 epitopes were predicted by the IEDB, MHC1 and 2 servers respectively, and B cells were predicted on ABC PRED. These epitopes were then tested for antigenicity, immunogenicity, non-allergenicity, and the conserved epitopes were selected. The CD4 epitopes underwent interferon gamma and interleukin-4 prediction, and population coverage was determined for HLA alleles that were associated with the epitopes. Our epitopes were docked to MHC1 and MHC2 complexes and validated using Peptide. And this led to our final design of the multi-epitope vaccine. The first result shows the HLA population coverage associated with our epitopes. So looking at the bars in green, which combines the MHC1 and 2 classes. This was important because these alleles tend to fluctuate between populations. And for a vaccine to be effective in lots of populations, you need a lot of them to be covered. So the combined alleles covered 89.4% of the world's population, which is quite promising. The highest coverage was recorded in Europe and North Africa, and the lowest was in Central America. Taking a closer look at some places that are still experiencing the burden of malaria, we can see that several of the alleles are included in this population. The coverage in SA is promising, which is the second graph, but may be an underrepresentation due to limited data for these regions. This is good because T cell receptors in a patient will easily recognize vaccines with multiple HLA alleles. These are the designed CD4, CD8, and B cell epitopes. These need to bind to MHC class 1 and 2 molecules to initiate the immune response. So CD8 is your cytotoxic T cells and CD4 are the helper. Here we have the CD8 epitope successfully bound to HLA. The epitopes are the colored strings in the middle of the diagram. We know that a more negative binding energy indicates stronger binding affinity. Our values range from -101 all the way down to -178. Similarly, for CD4, the epitopes bound to HLA. Energy ranged from -110 once again to 175. Interestingly, the strongest binding CD8 and CD4 epitopes are conserved, which is something that will be explored further. Lastly, docking of B cell to toll-like receptors is still to be conducted, as I ran into some issues with this. So this is an image of one of the toll-like receptors, which is TLR4. And this is necessary because the interaction between the toll-like receptors and the B cells is required for the immune response and is involved in lymphopoiesis of the B cell. So in conclusion, we can see that immunoinformatics is a very valuable tool in vaccine design. and the study successfully designed peptide vaccine. The epitopes were successful due to a high binding affinity, but this is just the beginning of a long study. The study needs to be further validated through synthesis and administration of the vaccines. These are some key references that I used, and we'd like to acknowledge the University of KwaZulu-Natal, CHPC for their service, and the National Research Foundation. Thank you all for listening.
Thank you, Leah, for a great presentation. Do we have any questions from the audience? Okay, I'll give everyone a few seconds. What would be the next steps in this research in order to end up with a vaccine?
There's still a very long way to go. So what I'd like to do from here, I can see someone say that Moses lab is the best. What I'd like to do from here is try and get into synthesis, which is quite costly, but it'll have to be necessary. So we're looking at synthesis and maybe some animal testing.
Okay, great. Thank you. We have another question. How widely are African populations represented in the MHC database?
From what I've seen and what I've read, a lot of people say that the African populations are underrepresented because a lot of data collection for that type of information is not being done in Africa. What really skews our results, especially when we're looking at something like malaria, which is predominantly in the Southern Hemisphere affecting Africa. So the safest way we can gauge it is to just assume that we are underrepresented.
Okay, great. Thank you. Here's another question. You mentioned using I can't say that word if you can see it in the, the comments. Would this be transferable to P, Vivax, and other malaria strains?
It should be, but I would suggest redoing the protocol with those strains to see if there is any difference. Because the problem with Plasmodium is there's a lot of variation genetically, and they have a lot of ways to escape the immune response. So because of that, I would suggest looking at each species independently.
Thank you.
I just chose Plasmodium because it's the predominant.
Okay, great. Looks like we have one more question. Are you doing the bench work on this?
Yes, we are. Hope maybe by next year we're hoping to recruit some MSc and honor students to go into it, because this is not my main PhD project, but a side project. So
okay,
it would be interesting to see it being carried on.
Okay, thank you so much. I don't think there are. Oh, yeah, I don't think there's any more questions. So we can move on to the next speaker.